Pioneer Award on Allen Institute Distinguished Investigator, etc., etc. She's a huge, incredible CV. So it's our pleasure to introduce Dr. Professor, actually, Mandë Holford. So please, my guest, Professor, the word is yours.
Everyone, and I'm happy to be here with you today, even though it's kind of virtual. I would have preferred to be in Brazil for real. Oh, I stopped sharing. Let me start it again. I hit the wrong button. Now, do you see the screen? Yes? Okay, great. So thank you for the introduction, and thank you, Marina, for the invitation to be here. Like I said, I wish I could have been in Brazil, but it's a little bit hectic right now with the end of the year in New York, but I'm happy that I'm able to participate in this way. And so today what I was going to share with you a little bit is how our lab has been working on venom peptides and sort of this transition, or I should say incorporation of new kinds of peptides. We mostly have worked on snails for much of my research. I've worked on venomous marine snails, cone snails, terebrids, and we've identified peptides that are used both in predation and defense from these animals. And as you know, and everyone here at this meeting knows, these arsenal that these animals are making have very, very important contributions to science, both foundational and translational contributions. And so now we're on this sort of purple venom trail as we work on other mollusks, and we're working now with squids, cuttlefish, and octopus, who also produce venomous compounds in specialty glands, mostly for predation. And so that's the story I'm going to share with you a little bit today. I have to show you a video of our killer snails, which you might have seen before. This is what I call the conidaean class of snails. As you know, cone snails produce conotoxins. The proboscis is the tongue. On the tip of the tongue, it has a harpoon filled with venom that it will then inject into the fish, and then it will have itself a meal. I never get tired of seeing this video. I absolutely love it. Whenever there's a hard day in lab and I think I can't do something, I play this video, and I was like, if a snail can eat a fish, I can certainly, purify a peptide. And so this is why we keep doing it. So while a lot of people are aware of, cone snails and terebrids, not so many people are aware that cephalopods are also very good predators, and that they also have toxins that they're using to paralyze their prey. So this is a cuttlefish. The cuttlefish will, excuse me, feed on the shrimp that was dropped into the tank. And first it will reach out its tentacles, grab onto the shrimp, and then inside of its buccal mass is where it envenomates it. And so what we want to know, the big question the lab is pursuing now, are how are these venom genes weaponized? So the assumption is that these venom peptides that we work on the genes that produce these peptides were co-opted from housekeeping genes and then became weaponized to be able to act the way that they do, to be very disruptive in cellular physiology when they are injected into predators and prey. And so what we're trying to figure out is, how did this weaponization occur? And so why this is important is because venomous animals, as you know, are found all throughout the tree of life, right? Almost every branch of the tree, everywhere you see red, you'll see venomous animal. And nature doesn't repeat a pattern over and over if it doesn't work. We just saw a snail eat a fish, you saw cuttlefish eating shrimp, and you all know the stories of snakes and scorpions and spiders. And so this is a very successful pattern that's convergent evolution that's occurred over and over again. And so it's a very nice model for us to understand a lot of things, not only about how biodiversity and speciation occurs, but complex trait development and novel systems. How are they doing? How do they form? And of course, now I think is the absolute right time to do that, right? So why this is important, like I said, and as you guys know, we've used these venom peptides to help us to map really important things, such as mapping the neuronal circuit of the brain using tetrodotoxin and bronchotoxin. And then also from these animals, either their peptides themselves or derivatives of their peptides have been used to develop drugs, right? So all of these are drugs that you can go out into the market and currently purchase. And then the most recent phenomenon that's driving everybody crazy is the Ozempic drug, which also came from the Gila monster. So not only have these peptides proven themselves in an ecological setting, they've also proven themselves in a translational therapeutic setting. And so that's why I think this meeting is so excited, because we're all looking to how we can exploit and leverage what we know to find out more ways that these peptides can be more quickly translated into therapeutic drugs. Why now is because I think we've gone from this, right, to this. So the amount of data that we can get from these animals in terms of what's in their venom arsenals, the structure of the venom arsenal, how the genes are homologous to other genes, all of that has sort of like exploded. So we are at a place where we can get so much more data, so much faster, and things like AlphaFold, which allow us to not only look at linear peptide sequences, but try to predict in 3D what they look like, have expanded our possibilities. And so it's important that we understand the technology that's coming at us and figure out how we can use it effectively. Another reason now is because we're in a crisis. There's a biodiversity crisis. We work with biodiverse organisms. We want to protect them. And there's a motto at my school, the care of the future of mine. Every peptide scientist, every biodiversity scientist should not only be thinking about what's the peptide that I'm going to get from this venomous animal, but how do I preserve the ecology of this animal so that we can learn more and continue to learn more. I think the stat is that we have over 200,000 venomous animals that are known on the planet, and less than maybe 2 or 3% of them, their venom arsenals are fully characterized. And so there's a lot of work to be done, but we can't do that work if we lose the animals, right? And so in my lab, we study venom evolution using a comparative phylogenetic method. We then try to identify new venom peptides and characterize the function of those peptides, mostly on ion channels and receptors at the surface of cells, in the hopes of identifying druggable compounds that might lead to treatments in pain and cancer. And so, and the newest thing we're working on, which I wanted to share with you a little bit at the end today, is how we're developing new venom model systems for doing this work. So today I'm going to talk to you a little bit about how we discover new venom peptides, and then also how we're making these new venom model systems using cephalopods. So as I explained before, most of my research has focused on conidaean snails, which are made up of cone snails, terebrids, and turids. I did my postdoc with Toto Oliveira, who I call the godfather of snails, and much of his work focused on cone snails and conotoxins. So when I started my own group, I focused then on terebrids and teratoxins. And so the traditional way that you used to find these snails is you'd go to the beach and collect as many of them as possible. And that's because that's what the technology allowed, right? You needed lots and lots of material. So size and quantity is what dictated which kinds of snails we studied. When I started, I wanted to use nature to guide us, right? So I wanted to figure out, using this phylogenetic driven genomics method, how was one snail related to the other, and how do their venom arsenals compare, right? So if we have this knowledge, it makes sense that the things that are grouped together on one branch might have similar venoms, while the ones that are distantly related would have different venoms. So in this way, evolution and diversity guided us in deciding which terebrid snail we would study, and we can do more target-driven discovery of what's in their venom arsenal. And so this is sort of what we've been doing. So we started by making the first molecular phylogeny of the venomous snails. We then identified those that were understudied or looked interesting. We then looked at different peptides within their venom arsenals with the hope of identifying individual ones that had bioactivity on particular channels. And so one scenario, we found a peptide that seemed to have activity on liver cancer. So this is the liver, the purple is the tumor, the blue is the receptor, and the orange that's coming in is the teratoxin peptide that is very specific for the tumors. So in liver cancer, there are certain channels that are dysregulated in tumor cells compared to healthy cells. So we're looking for peptides that would specifically inhibit the dysregulated channels in the tumor cells and not target the healthy cells, as a way of finding a more specific treatment for cancer. Because as you know, currently the treatment that's on the market, whether it's chemotherapy or some of the other drugs, they're not very selective, especially in liver cancer, in targeting healthy versus tumor cells. So here's where we wanted to use some of the specificity of what the venom peptides can do to target specific receptors that are overexpressed in tumor cells in liver. And so it turns out that TRP channels are the channels that are most dysregulated in several cancers, and in particular in liver cancer. You have an upregulation of TRPC6, TRPV1, TRPV2, V4, and M6. And especially in the US, liver cancer is the fourth largest, let's say, killer for mortality rate. And the current treatment of serafenib only extends treatment for a very short time. It's not extremely effective. It's better than nothing, for sure. But we were hoping that we could tackle this problem using venom peptides from our terebrids to come up with something that would be a little more substantial. And so we identified this peptide, TV1. It's the only peptide so far that's demonstrated to have activity against liver cancer. It's made up of about 23 amino acids. It has 3 disulfide linkages, and it is a part of what you would classify as the M superfamily, depending on its cysteine connectivity. And so we tested TV1 in both mouse models and in vitro. So what you're looking at right here is the result from the mouse model, and where when we treated it, normal is shown in purple, and cancer, the liver cancer cell model we were using, is shown in green. And when we applied TV1, the normal cells were not as affected as the cancer cells, as that's what's shown here in that plot. And so now the search was to find what other terotoxins can we have that have this kind of activity on TRP channels that we'd seen. So we started this computational screening method where we took all of our terotoxin peptides, filtered them based on looking for things that had disulfide connectivity similar to what we found with TV1, then predicted their fold using AlphaFold so that we would have this library. Then once we had the library, we then looked at clustering the library so that we could look at different chemical classes. So instead of looking at one individual peptide, we can look at a class or a series of clustered peptides. And then once we had those clusters, from those, we went through the clusters to then look at molecular docking to dock the peptides to published structures for TRP channels that appear to be upregulated in liver cancer. And then through molecular dynamics, we've produced candidates that we think are predicted to be druggable candidates for treating, or let's say druggable might be too strong. Let's say active candidates first against TRP channels, right? With the potential for being druggable. And so this is what some of these clusters look like. So from terotoxin, starting with over 3,000 peptide sequences, we then fall into several of these clusters, and I'm only showing you a few of them here. So cluster 18 is made up of 108 peptides. Cluster 12, you can see that the structures are quite distinct in the different clusters. And like I said, this gives us a group, or let's say a class of peptides that we can work on, as opposed to individual peptides the way that we would do in the past. And this again is just an innovation through having access to AlphaFold. Why now is a great time to be working on venom peptides, because now we can literally go from 1D to 3D in a matter of hours, or even days if you have big complex things. We did the similar thing with all of the conotoxins that are available on ConoServer, because not all of those have structures. And similarly, now we have classes of conotoxins that we can then try to see if any of these are going to target TRP channels in the way that we would like for disrupting liver cancer activity. And so I show you all this because right now we have all of these omic tools and functional assays that can help us get to druggable targets with venom peptides. But I do think we are missing a very valuable piece of the puzzle, and that's basically the genetics, right? So like you and other labs around the world, we have, for the most part, focused on the finished product, right? We focused on the amino acids, peptides, and proteins that are in the venom arsenals. But I think that focusing on these gene products has led to a knowledge gap. It's basically, I like to describe it like we are experts at describing all the parts of the car. We know the steering wheel, the headlights, we can figure out the hood, all of these things. But we don't really know why the car works the way that it does, right? And so to understand that, we have to go back into the assembly line to understand how the car was put together. So this is where we can connect what we see with what we can't see. So basically connecting phenotypic and genotypic biology. In order to learn and go through genotypic biology, however, that's the only way we're going to get to this big question of how these genes are weaponized, if we're actually able to manipulate the genes, right? So Venom is a molecular innovation. And in order for us to understand how these peptides function, we really have to understand a little bit more about the genes that are being expressed and regulated to produce the peptides. Um, so currently we don't have any genetically tractable molluscan venom model systems. The snails that we work with, they are wild caught. You get what you get, and you don't get upset. We cannot breed them in the lab. And the gold standards for model systems in science, none of these guys are venomous, so not helpful for us. So we decided we'd have to make our own venom models. And so that's what the lab is sort of focusing on now. So staying with mollusks, but branching out to cephalopods, so squids, cuttlefish, and octopus, we're going to try to use comparative analysis of these marine mollusks to determine novelty in gene and tissue development, the specialty glands that make the venom compounds. And this way we can compare molecular mechanisms of regulation and expression of these bioactive compounds. And so there are two methods in which we're trying to develop these methods. One is we're trying to make organoids of the venom glands so that we can sort of have our own benchtop reproducible models, so that we don't have to, like with snails, depend on wild-caught organisms, but we can have these 3D representation of the venom gland in the lab. So we can stockpile them. I like to say, like, beans or toilet paper, like we did during shutdown. The other method we're looking to go through is to make genome-edited cephalopods. And this is only possible because initiatives at the Marine Biological Laboratory, they're culturing these cephalopods, so squids, octopus, and cuttlefish. And now, because we have the genomes, we can identify specifically where on which chromosome a peptide is being expressed, or the gene that is expressing the peptide is located, go in and knock it out, and then we would have our sort of designer arsenal within these animals to study venom expression and regulation. And so if we're able to achieve these two things, I think it will radically, really change the way that we generate, manipulate, and utilize venom arsenals, while providing us that knowledge gap that we have to fill on understanding really how these peptides are working. So as a quick update on the venom gland, organoids, after lots of toil and trouble, it seems to be working. This is an organoid that's been alive for about 2.5 months in the lab. And then we did some HCR, in situ hybridization staining, and identified that it is expressing toxin. So it was a very joyful day in the lab after lots of toil and not so joyful days. But the one I wanted to focus on was on the cephalopod project, because this, I think, really is an innovation for how we can study venom peptides and their function. So Woods Hole is in Cape Cod, Massachusetts, and there's this place called Pie in the Sky, which really is like manna from heaven. So my being a Whitman Fellow there, to me, was really manna from heaven. And it's where I was introduced to and started working with these cephalopods. And like anyone that has a new area, you need teachers. So my octopus teachers were Roger Hanlon, Carrie Albertine, Brent and Taylor, who were the ones helped working with Carrie and Josh to develop these mariculture cephalopods. And then we had other people who are in the lab, and Lisa Abo is the director of the center. So all of them were very instrumental in helping us to learn more about the cephalopods and how they're producing their venom compounds. And so similar to snails, which produce their compounds in a venom gland, or snakes or spiders, cephalopods also produce in a specialty gland, and they produce in it's called the posterior salivary glands. And so it's located just right as highlighted here, just along the esophagus. They also have anterior salivary glands, but we didn't find much toxins in those, so we focused mostly on the posterior salivary glands. And so octopus have two of these glands, and cuttlefish tend to have two as well. In the squids, Dortoothus palei have one, and generally most squids only have one gland. But there is a species called Primna berrii, which is classified as a squid, but it does have two posterior salivary glands. So what we started was with transcriptomes, the same way we started with the snails, taking the glands out directly from the animals and making transcriptomes of them. And then the first thing we also wanted to do was to milk directly from the cephalopod. So this is a squid. We put it inside of a plastic bag. You can see the beak. It's not happy to be in the bag. So it's trying to bite its way out. And so when it does, it will releases this saliva, which you see there, which we then collect and we put into a functional assay using Artemia or sea monkeys. So without the extract, they're swimming pretty happily. Once we've added the extract to the animals, you can see that their swimming is now restricted and eventually they die. So this is our confirmation that yes, indeed, the saliva does contain toxic compounds, and this is published in literature from Lo Bianco, showing that octopus, cuttlefish, and squids can paralyze crabs and other things. So we built this database using both public and in-house generated transcriptome data from about 9 different species. We used 4 different squids, 3 different squids, 2 different cuttlefish, and then 4 octopus that were available. And so what we found is that a lot of the activity that has been reported, they're paralytic, they're hemolytic, coagulatory, and there are a lot of digestive enzymes. Of course, it's the salivary gland. And so the peptides and proteins we've identified mostly are bigger proteins. And the thing that's common to most cephalopods is that they produce something which is the name cephalotoxin, right? And so cephalotoxin is found all throughout, across taxa. And the first one to identify came from a squid, and it's SE cephalotoxin CTX. But we found it expressed throughout E. varei, palei, vandensis, and officinalis. We found it throughout all of the cuttlefish and squid samples that we tested. Did not find this particular CTX in octopus, however. And what's interesting is there is homology among the organisms, but the sequence itself is not exactly the same, right? So in squid and cuttlefish, you'll find CTX, but like I said, in the octopus, we did not find homologs to the Sepia elondanus CTX, which was originally reported in. And the only one for which we have a complete sequence of what is the venom compounds in these cephalopods. And so what we wanted to know, now that we'd identified this CTX that we can see is universally being expressed across different taxa, is what can we learn about the development of the gland and also the expression of the gene. So this is a development chart from Cuttlebase. Tessa Montague mapped this out for cuttlefish, where you can go from, embryonic stage 1 all the way to hatchling at stage 25. And this is the power of having a venom model, right? Because now we would be able to trace the development of the venom gland, and we can also trace the development or the expression and regulation of the venom compounds as well. So we wanted to know, when does development of the posterior salivary glands start? Do all the lineages start at the same stage? And then also, when do they start to produce their venom peptides? When do they start to produce the venom compounds? And so in doing this work, we started with E. varei, which is the squid, and we identified that it's mostly late stage. So the squid staging is a little different than the cuttlefish staging. But at stage 26, 27, we were using in situ hybridization probes that were specific for the gene transcript for CTX. And so what you're looking at here is basically a baby squid. These are the arms over here. These are the eyes, and that's the rest of the body. And so where we're expecting to see the posterior salivary glands, remember barei has 2, would be around this area. And we see that when we get into later stage. So right here, these two little red things that are very overexpressed, those are the areas where you're seeing the CTX gene within the whole mount of this um, organism. We then did sections of the hatchling once it's hatched, and using the same CTX in situ hybridization probe, we can clearly see that the venom compound is only expressed in the posterior salivary gland. And again, they have a left and a right gland. What we originally was looking for was if there were differential expression between the left and the right. It doesn't appear to be so. It seems like they are equally expressing the CTX, although I'll talk a little bit later about where we are seeing differential expression in these glands. So this again was like, oh my God. So for the first time ever, we've been able to highlight, trace, number one, the development of the gene, and number two, spatially visualize where in the gland the genes are being expressed. And so then we did this not just for baryi. So here, this is just looking at the posterior salivary gland itself. And as you go from left to right, it's increasing in magnification. And so we also did this for Sepia bandensis, the cuttlefish. So we've done it for squid, cuttlefish, and octopus, and you see the same kind of specific labeling only in the posterior salivary gland. So the venom compounds are not being made in other parts of these animals that we've analyzed. It's specific to the salivary glands. And so again, talking about differential expression, as you know, cone snails and assassin bugs have been reported to do this sort of differential expression where they have different parts of their glands or different glands in general for expressing both predative and defensive venoms. And so that our initial thinking was because these cephalopods in general tend to have two glands, that one would be a predative gland, one would be a defensive gland. We're not sure if that's the case because we saw, again, CTX was expressed in both glands. But if we're able to identify this kind of differential expression of the compounds, it will help us to determine key factors involved with the emergence, proliferation, and differentiation of the genes. And so we do see some differential expression, not in left and right, but more spatially within the glands, right? So again, this is Palea, it's a squid. In green is the CTX in situ probe that we've been using. And in red is another venom compound that we've identified that's highly expressed in this gland. It's an unknown compound. We don't know where it's from. We don't know what it does. We know where it's from. It's similar to what was found in an octopus by Mycoloides, but you can clearly see that the DP PT2 peptide is mostly on the peripheral of the gland, whereas the CTX seems to be expressed mostly inside of the gland. And so we've done that for others and shown that there is differential expression also in octopus and in cuttlefish. And so now we feel like we have a handle on at least CTX. What would it be if we can go on and knock out the CTX gene and see how it affects development of the gland itself and also development of the venom arsenal. So like in snakes, you know, there's the dominant toxin theory. And so if you knock out that dominant toxin, what else is going to be produced, right? And we can do this again because we have genomes of all of these different organisms. Some things we do not have when we're working on the snails. There are only about 2 or 3 reliable clone snail venoms to the chromosome level. There are none for terebrids. We've been trying so hard, but it's just so difficult. You can't get enough material, but we keep trying. But here we already have all of these genomes. And so again, we can locate a specific loci on a chromosome that's expressing the CTX, knock it out, and see what happens in terms of development. So we can make these genome edited models to determine more about how these genes are being expressed and regulated, right? Which will then lead to determining the form and function of these compounds. So we've just done this experiment where we've produced guides. We're using CRISPR-Cas9 system. We've produced these guides. We're doing this in collaboration with Carrie Albertine from MBL and Tessa at Columbia. and now we're sort of crossing our fingers and hoping that they hatch, and then we'll be able to see what happens in terms of development and expression of these toxins. The last thing I wanted to just touch on before, and if I'm going long, please tell me and I'll stop. The was about how the venom is being released, right? So this is a recent review that talked about how venom is released in these different kind of animals. And for mollusks, here at the bottom, you'll see the little snail. It's predicted that they'll have this holocrine released where the cells sort of rupture. Not sure, we want to find that out. There's no obvious mechanism for posterior gland contraction in cephalopods like snakes. As you know, have this big musculature tissue that they use to then pump their venoms. Our snails have a venom bulb, which is basically a big muscle that pumps the venom through the venom duct. And then cephalopods, on the other hand, don't have a big muscle that they can identify, but there was work done that identified that the release might be under neuronal control from the superior buccal ganglia, which then feeds into the posterior salivary gland nerve, which runs along the salivary duct, and then releases into the muscular salivary papilia, which then injects the or releases the venom. And so we wanted to test this to try to look for what kind of muscular fibers might be present in the salivary gland. So using phalloidin, which is an acting muscle fiber staining, we're able to see that all of these, within the venom gland, in the salivary glands, you have a lot of phalloidin there, so it is possible to get muscle contraction of the individual cells. And then there was a paper by Young in '65 where he basically ablated the nerves that are responsible for venom release, so the salivary nerve, the interbuccal nerve. So what he took 10 different octopus, that's what you're looking at, the MC whatever is the name of the different octopus, and half of them he did nothing to, positive, and the rest he cut the nerves, that's the negative, right? And then once he did that, he observed their behavior. For the controls, they behaved as normal. They would poison the crab, clean the crab, and then or eat the fish. But for the ones in which he had cut the nerves to the salivary glands, you see that it didn't poison the crab, clean the crab, or eat the fish, suggesting that neuronal control is a very important key piece of how their cephalopods are releasing their venom. And so we wanted to visualize the nerve connections in the posterior salivary glands and then identify possible ion channels or receptors that might be related to the muscle contraction in the glands. So we did some neuronal staining using pan-neuronal dyes. This is Sanoff, which stains synapses. So you're looking at the brain and a tentacle for this cuttlefish, Pandansis. And so in the brain, it lights up because there are lots of synapses. And then when we looked at the posterior salivary gland, in this case, this is from Palei, also got very marketed, distinct staining. So again, you see the Sanoffs are all along, and it seems to be on the membranes of the cells within the salivary gland. And then we used CHAT, choline acetyltransferase, which then seems to also indicate, so green is the cephalotoxin, blue is DAPI, and the red is the indication of where you're seeing the choline acetyltransferases are active. And so it definitely looks like there's innervation or within the gland of the acetyltransferase. And then we took this is in collaboration with Eve and Amy, we took the gland itself and soaked it in Cal 530 and then spiked it with dopamine. And so you can see firing happening a little bit here on the surface. And then what I think is kind of interesting is you see this little, I don't know if it's a bubble or something comes to the surface and then it gets released. So these are all really exciting times, and we're hoping to get to the bottom of this right now. So where do we stand? I personally think cephalopods rule if you want to do venom research and look at venom peptides and compounds and how they're being expressed and regulated. And so I just wanted to quickly add a little plug about sharing knowledge and how it can facilitate advances. There's a lot of work from Flo, Florence, and others who've set up the Venom Zone, which we all use, as well as ConaServer and Uniprot. These are common tools that we go to figure out if we have something novel or something known or how it works or where it works. And so this sharing has really helped our field to grow. And I think also setting standards would be great. So we've developed this Venomics training course that is available from Millard, in which it teaches the modules of transcriptomics, genomics, and proteomics for how we go through and do identify these transcripts that we all have from different organisms, the genomes that are now coming online, and the proteomes, so that we all are looking at the data and collating the data and identifying and naming things in sort of a standard way. Because once we have those kinds of pure standards, again, it helps us to leapfrog, right? So kind of like the idea is we want to set up a Venom base, kind of like Phi base or Zen base or Econobase. All of these databases have helped these fields to grow and really expand significantly. And so if we construct a Venom base, we can do something similar for our field. This figure was generated by Dolly, but I love it. And so it will really, I think, help us to drive Venom analysis if we're able to have a very curated and well characterized Venom base in order for us to look at these methods that we're all starting to use, these methods and others, right? And so this is one, we've submitted a proposal for this. Fingers crossed, NSF likes it and they give us some money to make a Venom base, and then we'd be reaching out to all of you to ask you to share your data sets and whatever else you would like to share, because it would cover both phylogeny and systematics, and as well as functional data, and of course all of the omic data that we've collected. So with that, I hope that you can understand this sort of journey that we've been on from snails to cephs that encompasses all of the work that we've done for venom peptides and venom compounds. And why I really do think it's an exciting time is because we're able to have now these kind of model systems to investigate a lot more about how our peptides work and how they are expressed and regulated. And so like Da Vinci's model, we're hoping these models will help us to soar to new heights in the venom field. And with that, I thank you very, very much, and thank all of the people in the lab who are doing this work. Jose was a postdoc in the lab that started to characterize the CTX function. Sarab, Kim, also other postdocs that are working on the project. Belkis is a grad student who's done a lot of work in the lab, as well as Melvin, who's a technician. And so this is the crew with the ice cream that we took to the Marine Biological Laboratory last summer, who really helped us to get a lot in. And Jordan Wynn was a undergraduate REU student at Woods Hole that worked with us. And so I've mentioned the names of the people that's throughout the talk, and of course thanking the funders and thanking you for your attention. We don't bite, only the mollusks do. And so I'm happy to take any questions that anyone might have. Thank you very much.
Thank you very much, Dr. Professor Holford. This is very, very beautiful work, very nice. I actually start this questions. I start with a question regarding CRISPR-Cas9 system, because of course, I work with CRISPR-Cas. I know how hard it is to obtain these mutants. It's very difficult to obtain them. Of course, it's hard work. Be careful. Every single step is very challenging sometimes. So I was wondering about. I think it's the first time you do this in your lab. You're expecting to get a mutant soon. I hope, fingers crossed for you. So I was wondering about your model, because I'm totally outsider of this kind of research. So I was wondering how you make selection of these cephalopods and everything in your lab. How you do? Because I think you put them in rearing conditions to develop, etc. So how you select the mutants for this step now?
What's nice about the cephalopods is that you can buy them. It's like Amazon. So the Marine Biological Laboratory has set up a program where we can order, embryos, or we can order hatchlings, or we can order full adults. And so right now, we don't have an aquatic facility in our lab. When we're doing the injections, we work in collaboration with Tessa and Carrie at the MBL. And so we do the injections there with them, and then they'll grow the animals for us. And then once they hatch, they will then send them to us for analyses. So then we will then do the work
sequencing,
to
etc.
analyze, what the genotyping and all the other stuff to see what's working or what's missing or what's going on. And so it's very much a collaborative effort, but the only way it's possible is because we can order the animals directly from MBL. So it's a fee for service. And we don't have to do the husbandry ourselves.
Okay, thank you. So anyone has questions? No? Okay, Professor Holford, once more, thank you very much for joining us today. I hope to see you soon here personally with us. Okay, thank you very much.
Thank you very much, and I hope the rest of the meeting goes well. Have a great time.
Thank you very much. So, the last guest of our session this morning, I'd like to introduce